You glow in the dark. You glow in the dark. Not metaphorically, not as a warning display. When ultraviolet light touches your exoskeleton, your entire body emits a blue-green fluorescent glow that has puzzled scientists for decades because no other arthropod does this consistently, and nobody has yet reached a complete consensus on why. You're a scorpion, and the mystery of why you glow is only the beginning of what makes your species one of the most extraordinary survival stories in the history of life on this planet. You have existed in essentially the same form you wear today for approximately 430 million years. That number requires context to fully understand. 430 million years ago, there were no dinosaurs. There were no mammals. There were no birds, no reptiles, no flowering plants. The continents looked nothing like they do today. And yet, in the ancient shallow seas of what would eventually become dry land, something that a modern scientist would recognize as a scorpion was already hunting, already using venom, already raising live young, already glowing under the light spectrum that you glow under today. Cornell University has documented the fossil record of scorpion evolution extensively, tracing the lineage from marine ancestors in the Silurian period through the transition to terrestrial life, describing it as one of the most stable body plans in the entire history of animal evolution. The Smithsonian Institution holds fossil specimens of ancestral scorpion species that demonstrate the same basic architecture you carry right now. The segmented tail, the claws, the stinger, preserved in rock hundreds of millions of years old. When evolution finds something that works, it tends to keep it. You are not an insect. This matters, and it is worth stating clearly from the beginning. You are an arachnid, a member of the same class as spiders, mites, and ticks, distinguished from insects by your 8 legs rather than 6, and by a body plan that connects your cephalothorax and your abdomen without a waist in the way a spider has one. Your relationship to insects is roughly the same as a human's relationship to a fish. You share an ancient common ancestor, but the lineages diverged so long ago that your similarities are those of deep structural origin rather than close kinship. What you share with the insects of this field is something more fundamental. You share a planet, a history, and an ecological role as predator, prey, and participant in some of the most ancient living systems on Earth. You hatch inside your mother's body. In most scorpion species, eggs are fertilized and develop internally, and the young are born live, fully formed, tiny white versions of the adult that will climb immediately onto their mother's back and ride there for the first week to 2 weeks of their lives while their exoskeletons harden and darken. Your mother does not leave during this time. She does not hunt. She positions herself in a protected location, stands still, and carries you and your dozens of siblings on her back until each of you is ready to disperse. If you fall off, she will often reach down and guide you back. If a predator threatens, her defensive response is immediate and total. The National Geographic Society has documented maternal behavior in scorpion species across multiple desert ecosystems, describing the relationship between mother and offspring during the first weeks of life as more behaviorally complex than most people expect from an arachnid, involving active repositioning of young, aggressive defense of the brood cluster, and in some observed cases, deliberate feeding behavior where the mother presents prey to offspring still riding on her. Entomologists and arachnologists at the Entomological Society of America have spent decades studying scorpion reproductive strategies, noting that the investment a female scorpion makes in each reproductive cycle, the internal development of young, the extended carrying period, the defensive posture during that window represents one of the highest levels of parental investment seen in any arachnid species, and rivals or exceeds the parental investment of many vertebrate animals people intuitively think of as more attentive parents. After leaving your mother's back, you are on your own. You will molt multiple times over the next several years, shedding your exoskeleton as you grow, each molt leaving you temporarily soft and vulnerable in the hours before your new shell hardens. During those vulnerable hours, you do not move, you do not eat. You find the most sheltered location available and you wait, because the soft stage between molts is the period of your life when predation risk is highest. You reach maturity slowly, Most scorpion species require 2 to 6 years to reach reproductive age, a timeline that would seem extraordinary for an animal. Most people categorize mentally, alongside insects, as something small and short-lived. The lifespan of a scorpion in the wild ranges from several years to over a decade in some species, with captive specimens of certain species documented living for 25 years or more. Let's establish something about how you hunt, because it is the foundation of almost everything else worth understanding about your biology. You are primarily nocturnal. You spend your days in burrows, under rocks, beneath bark, in crevices, conserving water, avoiding the heat that would kill you in hours if you were exposed to it. When darkness falls and temperatures drop, you emerge and begin hunting. Your sensory system for locating prey in complete darkness is a masterpiece of evolutionary engineering that researchers are still fully mapping. You have up to 12 eyes depending on species, including 2 large central eyes and rows of smaller lateral eyes along the front of your cephalothorax. But your eyes are not your primary hunting tool. In low-light conditions, they primarily help you orient to moonlight and starlight. Your primary sensory organs are the pectines, comb-like structures on the underside of your abdomen that detect chemical signals and vibrations in the substrate beneath your feet. These organs allow you to detect the movement of prey through substrate vibrations, to orient toward chemical signals in the air, and to navigate terrain with a precision that does not depend on visible light at all. The University of Arizona has conducted detailed research on scorpion pectine function, documenting how these organs can detect substrate vibrations produced by prey moving as far as 50 centimeters away from the scorpion's position, providing a directional sense of prey location before any visual confirmation is possible. You also have slit sensilla, microscopic sensory structures distributed across your body that detect air movement with extraordinary sensitivity, allowing you to sense the wing movements of flying insects approaching from any direction in complete darkness. In a dark desert environment, you are not hunting blind. You are hunting with a sensory package specifically optimized for an environment where visible light is absent and prey is detectable through vibration, chemistry, and air movement alone. Let's reconstruct a hunt. It is 3 in the morning. The desert air has cooled to a temperature where your metabolism is efficient and your movements are precise. You have emerged from your burrow under a rock that holds daytime heat, and you are positioned on a flat area of sand, facing into a light breeze, your pectines pressed against the ground. Something moves 20 centimeters to your left. The vibration signature is consistent with a cricket, medium-sized, moving across the sand surface at irregular intervals. You pivot without lifting your body. Your legs reorient you toward the source of the vibration. You begin to move in short, controlled steps, pectines continuously reading the substrate, constantly updating your position relative to the prey. You close the distance to 5 centimeters. The cricket has not detected you. Scorpions are among the quietest hunters on land, moving with a controlled precision that minimizes substrate disturbance even at close range. Your claws, called pedipalps, shoot forward and close around the cricket's body before it can react. Now, the decision that separates scorpion venom biology from almost every other venomous animal on Earth. You have venom. Your stinger, the telson, curved at the end of your tail, contains a venom gland capable of delivering a sophisticated mixture of neurotoxic compounds, enzymes, and peptides that can disable prey almost instantly. But for a cricket, a prey item small relative to your body size and already immobilized by your grip, you do not use it. The University of Exeter has documented scorpion venom use in prey capture extensively, finding that most scorpion species use mechanical restraint alone for small, manageable prey, reserving venom injection for larger, more dangerous prey items that struggle effectively enough to risk injuring the scorpion's claws, or for defensive encounters with predators too large to hold mechanically. This selective venom use has a physiological basis. Venom production is metabolically expensive. The body must synthesize the complex protein mixture from scratch, and replacing a depleted venom gland takes time. A scorpion that expends venom on every prey encounter will eventually face a predator it cannot defend against because its venom gland is empty. You grip the cricket. It struggles briefly. You begin to feed using your chelicerae, the small claw-like mouthparts hidden beneath your claws, to pull food toward your mouth, processing the prey with a combination of external digestion and mechanical grinding. This is the daily reality of scorpion life in most environments. Patient sensory monitoring, precise nocturnal hunting, selective use of the extraordinary biological weapon at the end of your tail. Now let's talk about what that weapon actually is, because the venom of scorpions is both more varied and more medically significant than almost anyone outside the research community fully understands. Across the 2,500 described scorpion species on Earth, venom composition varies enormously. Most scorpions produce venom that is mildly to moderately painful to humans, but not life-threatening. Most stings feel like a severe bee sting or wasp sting, causing localized pain, swelling, and temporary numbness. But approximately 25 to 30 species produce venom capable of causing serious illness or death in humans without medical treatment. These species are disproportionately concentrated in the family Buthidae and include species found across North Africa, the Middle East, South Asia, and parts of the Americas. The most dangerous species by venom potency is widely considered to be Leurus quinquestriatus, the deathstalker scorpion, native to the deserts of North Africa and the Middle East. The Royal Entomological Society and the National Institutes of Health have both published extensive analyses of deathstalker venom composition, identifying a complex mixture of compounds including chlorotoxin, agitoxin, cytotoxin, and cryptotoxin, each targeting different components of the nervous system with remarkable specificity. Chlorotoxin, one of the most studied compounds in any animal venom, binds selectively to a specific type of chloride channel found in tumor cells. This binding property, discovered in the 1990s, has made chlorotoxin the subject of intense pharmaceutical research as a potential cancer diagnostic and therapeutic agent. Researchers have used modified chlorotoxin molecules to create tumor-targeting agents that can identify cancer cells with precision that standard imaging sometimes misses, because the cells express the chloride channels that chlorotoxin recognizes with far higher frequency than healthy tissue does. Agitoxin and cryptotoxin, meanwhile, block specific potassium channels in nerve cells, interrupting the electrical signaling that allows muscles to function properly and the nervous system to regulate heartbeat and breathing. In high enough doses, this interruption causes the cascading cardiovascular failure that makes untreated envenomation by dangerous scorpion species potentially fatal. The counterintuitive element of scorpion venom biology that surprises most people is the specificity of these compounds. You are not injecting a general poison. You are injecting a pharmacological library, dozens of distinct molecular compounds, each of which targets a specific receptor or channel in the nervous system with a precision that pharmaceutical chemists spend decades trying to replicate artificially. Nature has published landmark research documenting how scorpion venom has been the source of more biomedically significant molecular discoveries per species studied than almost any other venomous animal group, with compounds from scorpion venom now in clinical trials or active pharmaceutical development for pain management, cancer treatment, autoimmune disease, and cardiovascular medicine. You do not know any of this. You have never encountered a cancer cell. You built these compounds under evolutionary pressure across hundreds of millions of years, optimizing them for prey capture and predator deterrence, not medicine. The pharmaceutical applications are a human discovery layered onto a biological system that was never designed with human benefit in mind. But the precision is real, and it is extraordinary. Let's talk about the glow, because it is one of the most visually striking features of your biology and one of the least understood. When ultraviolet light, wavelengths shorter than visible light and longer than X-rays, strikes your exoskeleton, specific compounds within the outer cuticle layer fluoresce, absorbing the ultraviolet energy and re-emitting it as visible blue-green light. The compounds responsible are primarily beta-carboline derivatives and certain coumarin compounds that accumulate in the hyaline layer of your cuticle, the outermost transparent layer of your exoskeleton. This fluorescence is so reliable and so strong under ultraviolet light that researchers use ultraviolet flashlights to locate scorpions in the field at night, with an effectiveness that far exceeds searching by visible light alone. The question of why you glow has generated multiple competing hypotheses in the scientific literature. Some researchers at the University of California have proposed that the fluorescence functions as a moon-sensing mechanism, helping scorpions evaluate ambient light levels and decide whether to remain active or return to shelter. Scorpions are known to retreat from bright environments, and the fluorescent compounds in the cuticle may allow the scorpion to sense the intensity of its own glow as a proxy for ambient ultraviolet exposure. Other researchers have proposed that the fluorescence attracts or disrupts insects that are sensitive to ultraviolet wavelengths, potentially functioning as a hunting aid by drawing prey closer. Others have suggested it may be a byproduct of a cuticle hardening process with no direct adaptive function at all. What is agreed upon is this: the fluorescent compounds appear within the cuticle immediately after each molt, as the new exoskeleton hardens. Newly molted scorpions with soft, unhardened cuticles do not fluoresce. As the cuticle matures and hardens, fluorescence develops. This suggests the compounds are integral to the cuticle structure itself rather than separately produced. Cornell University has proposed that this relationship between cuticle maturity and fluorescence intensity may actually give scorpions a passive ability to monitor their own exoskeleton condition, with brighter fluorescence indicating a fully hardened, fully protective cuticle, and dimmer fluorescence indicating a recently molted, still vulnerable state. Whatever the function, the glow exists. It has existed for as long as scorpion cuticle chemistry has existed, and it remains one of the most beautiful and most mysterious features of a biology full of both. Let's talk about what you can survive, because this is where the story of scorpion resilience becomes genuinely difficult to believe without the research to anchor it. You can survive being frozen at temperatures below 0 degrees Celsius and return to full activity when thawed, provided the freezing happens slowly enough to allow your cells to adjust. Researchers have documented this in multiple desert scorpion species, where overnight temperatures can drop far below freezing in certain seasons. The scorpions enter a state of suspended activity and resume normal behavior when temperatures rise. You can survive submersion in water for hours, entering a state of metabolic suppression while submerged and resuming activity when removed. This capacity likely relates to your evolutionary origin as a marine animal, retaining certain physiological responses to aquatic conditions hundreds of millions of years after your lineage left the sea. You can survive extremely low food availability. Certain desert scorpion species have been documented surviving on a single insect meal per year under laboratory conditions, slowing their metabolism to a rate that allows energy storage to sustain all critical functions for months without additional input. Your metabolic efficiency at low activity states is among the highest recorded for any animal of your complexity. The University of Florida has published studies on scorpion metabolic suppression during food deprivation, documenting survival periods and metabolic rates that have informed research into extreme physiological adaptation in other animals, including work on dormancy mechanisms in mammals and torpor responses in reptiles. You can also survive radiation exposure at levels that would kill a human within hours. Nuclear testing sites where ambient radiation levels remain elevated have been documented hosting scorpion populations, and laboratory tests exposing scorpions to radiation doses. Multiple times the lethal dose for humans have produced scorpions that survived and continued normal behavior. The mechanisms behind this radiation resistance are not fully understood, but are thought to involve the efficiency of your DNA repair systems and the structural properties of your exoskeleton in deflecting certain types of radiation. What does it mean to have survived 430 million years? It means surviving the Permian-Triassic extinction, the largest mass extinction event in Earth's history, which killed over 90% of all species on the planet. It means surviving the event that killed the dinosaurs. It means surviving the ice ages, the continental shifts, the atmospheric changes, the rise of humans. Whatever conditions produced on this planet, your lineage found a way through it. This is the part of the story where the question of resilience becomes something more than biological. What does it actually mean to persist through conditions that erased almost everything else? What is it about the scorpion's particular combination of characteristics, the sensory precision, the metabolic flexibility, the defensive venom, the conservative reproductive investment, the behavioral patience, that made this body plan durable enough to survive everything Earth has thrown at living things for nearly 500 million years? Researchers who study evolutionary stability across deep time consistently point to the same cluster of traits in scorpions. Generalism in diet. You eat almost any arthropod or small vertebrate you can subdue. Behavioral flexibility. You adjust activity patterns, reproduction timing, and shelter use in response to environmental conditions. Physiological range. You tolerate temperature extremes, dehydration, food scarcity, and environmental chemical stress far beyond what most animals can endure. And reproductive investment. You produce fewer young, but invest heavily in each one, a strategy that proves durable across environmental change. Does that combination sound familiar? The organisms that survive through the deepest periods of change in human history are rarely the most dominant, the most specialized, or the most visible ones of their moment. They are the ones that combine enough flexibility to adapt to what changes with enough core function to maintain what matters. The ones that can wait out the worst conditions and resume when conditions improve. The ones that carry something in their biology or their strategy that remains useful regardless of what the environment demands. The scorpion did not survive because it was the most powerful predator of any era it lived through. It survived because it was consistently useful, consistently adaptable, and consistently willing to wait. You glow in the dark. You emerged from ancient seas 430 million years ago, and you are still here, carrying venom that modern medicine is only beginning to understand, raising live young on your back in a desert that would kill most animals in a day, hunting in complete darkness with sensory systems so precise they detect prey through vibrations in the sand, surviving conditions that have ended entire branches of the tree of life. The dinosaurs are gone. The trilobites are gone. The vast majority of every species that has ever lived on this planet is gone. And yet tonight, in a desert somewhere, when the ultraviolet light touches the sand, something glows blue-green in the dark, standing perfectly still, reading the vibrations beneath its feet, waiting with the particular patience of something that has survived everything before and has no particular reason to believe this moment will be any different. You glow in the dark, and somehow, after 430 million years, you are still here to do it.